Blown up biology. For many years I've had a really strange obsession with stone walls. I find it impossible when I'm walking in the countryside or even in the town to go past a stone wall without staring closely at the stuff that's living on the stones. And it prompts all kinds of thoughts about the nature of life. What is it that makes stones turn green? Provided a stone is damp and is surrounded by air and has sufficient light, it turns green. Water, air and light. What I'd like to do is to show you the kind of organism that could exist in a very small pool of water. Perhaps at the foot of a part of a sunlit wall, or a hollow in a stone. And bear with me. Here I have a microscope slide, and earlier this morning we heard about Euglena. I'm going to show you some live Euglena. And we heard how Euglena can swim. The more brightly lit side of this file of Euglena culture is green, and the back is clear. I put a small drop on the slide of this greenish coloured water. Now you can see this during the warmer months in the countryside, in the town, but especially in a cattle trough where cattle have been drinking. Very often the water goes bright pea green. I put a drop on the microscope slide, on the glass slide. I'm going to put a very thin glass cover slip on top to flatten out the surface of the water. Let's have a look under the microscope. You might notice straight away that these things are moving. That's what they sound like. Let's go in a little bit closer. Now some of them are stuck to the glass slide itself, so if I can pick some of those, I should be able to zoom in even closer and see them even more clearly. Can anyone notice anything about the colour of these things? They're green and they're swimming. Does that strike anybody as being a little bit strange? They swim by means of a propeller. Life may not have invented the wheel, but it has invented the propeller. I don't have time to spend messing about with a microscope trying to set it up so you can see this thing. It's called a flagellum. It's a thin corkscrew-shaped spiral thread projecting forwards from the cell, and it's spinning. It's being driven by a molecular motor, spinning in a molecular bearing at the front end of the creature. I used the word cell just now. These things are single cells. Okay. And there's a clearer photograph taken under better conditions showing the flagellum. Now I'm going to show you a creature which consists of a number of cells. It's called Volvox, and it's extremely beautiful. These are quite common in ponds local to where I live. I was lazy and bought them from a biological specimen supply company. But if you see pond water which is bright green and has little pinhead-sized dots moving in it, you've found a colony of Volvox. And again, the front of this vessel of water is green and the back is clear. You can't see that, unfortunately. Well, you might be able to, actually. I don't think you can. No, I don't think you can see it. So, let's put some Volvox on this slide. Now, this slide is deeper and is marked with 1 millimeter graduations. Yes, you can just about see them. Okay. I'm allowing you to see me set these slides up because I think it makes it so much more real to see the scale and the reality of what we're looking at. Now let's look at Volvox under the microscope. Can you see them moving? If I can find one that's stationary, I'll be able to zoom in again. Those are 1 millimeter squares. Go choose. That's a nice specimen down there. Now, Volvox, oh, there's an even nicer one. Volvox is like a hollow football. Going closer. Beautiful. And it's a single layer of cells, rather like Euglena. Only each of these cells has 2 flagella. And inside you can see daughter colonies. Each of those is a hollow ball of cells. And Volvox reproduces simply by bursting open and releasing those daughter colonies out into the pond to grow separately. So now we have an organism which, instead of just being a single cell, is a number of cells living together. I'm going to show you another creature you probably all remember hearing about in biology classes if you ever did biology at school. It's called Spirogyra. That was my nickname when I was a biology teacher. Spirogyra, you've often seen in ponds and ditches. It looks absolutely disgusting. It looks like a great big bubbly mass of green slime. I was going to use a ruder word. It's horrible looking stuff. It's extremely slimy, extremely bright green. Here we have some. And if I pick it up, with the forceps, you can see it's slimy looking stuff, but actually it looks rather like hair. And that's not misleading, because it is very like hair. I'm going to put, again, a drop of water on the slide. It's always easier to see things in water with a microscope. And now I'm going to take out some Spirogyra, and because it's very long, I'm going to snip it. There we are. And put that in the water. Now can you see it really is like hair? Can you see that? Just about. We have to flatten out the surface of the water again with a thin glass cover slip. And we'll see what this looks like. It's remarkably beautiful. Let me get it in focus. Now you can see why it's called Spirogyra. It has very intricate spiral patterns. Let me explain what you're looking at there. You're looking at chains of cells, shaped rather like, well, in that case, baked bean cans, stacked one on top of the other, joined end to end. It's difficult to envisage the three-dimensional shape here, but it's a cylinder. Each cylinder is a separate cell. Let's go in a little bit closer. We've actually got quite a good specimen right there, so we'll zoom in. Please ignore the small organisms moving around. We're going to talk about those later. They're not part of our story yet. We're still focusing on these green things. You may just be able to notice a nucleus in the center of each cell. Now, we should be able to zoom in even closer. We can. And there. That's very clear indeed. That's good. And I can focus. If I focus down the bottom, the spiral is the oblique lines are running that way. If I focus on the top, they're running that way. That's a pretty clear demonstration that we're dealing with a three-dimensional spiral. The spiral is the green bit. We call it a chloroplast, and that's the part of the cell which is responding to the water, air and light in order to create life. It's producing food simply from water, air and light, and it's storing that food in the form of starch grains, which you can see sparkling there in the middle of the chloroplasts. Those are grains of starch. And in the center there is the nucleus. It's shaped a bit like a Smartie. It's a squashed shape, and it's suspended in strands of protein, which is rather like egg white, which also lines the cellulose walls of the cell, and the middle part of the cell is full of water. So can you picture the sort of sticky egg white strands stretching out to the egg white lining the edges of the cell and forming bridges to this Smartie-shaped nucleus that's suspended in the middle. It's absolutely beautiful, and certainly belies the rather disgusting image we have of blanket weed in a pond. So I hope next time you see that stinking, not necessarily stinking, but that slimy, noxious looking green stuff in a pond, you'll just think of the beauty of life. These are lichens growing on a stone wall. Lichens are remarkably tough organisms. They can survive in extremely dry environments. They're extremely beautiful. They're also an indicator of the cleanliness of the air. The more lichen species you see on a stone wall, the less atmospheric pollution there is likely to be. They're very sensitive to acidity in the rain. This looks like almost like a map, doesn't it? These things, they also remind me a little bit of a mouldy slice of bread. And the resemblance is not a coincidence, because they form in a very similar way. A mouldy slice of bread forms patches, because each patch has grown out from a single airborne spore which has landed on the bread slice. In just the same way, these patches of lichen grow on a wall. It's possible to estimate the age of a stone wall by looking at the diameter of the patches of lichen. And again, I find them fascinating to look at when I'm outdoors walking. Vegetable dyes were extracted from some of them. I think this species is one of them. Some of them are quite alien-looking shapes. Lichens are a symbiosis between a fungus and an alga. Now then, I'd like you just to think for a moment about what it would be like to live on a stone. Try and empathise with these creatures we've been looking at, and we're going to see more. I'd like you to just humour me and please close your eyes. Imagine that you live on a stone. Please close your eyes. Imagine that you live on a stone. You have nothing to survive from but water, air and light, and whatever might dissolve out of that stone, and whatever else might also be surviving on the surface of that stone. That's all you've got to live on, okay? Imagine how hard that must be. Now, open your eyes and just consider the fact that actually you do live on a stone. We all live on the same stone. It's a damp stone surrounded by air and bathed in light. And all we have to survive on is the water, the air, the light, whatever it is that dissolves out of that stone, and whatever else it might be that's also living on that stone. When I realised this, it suddenly dawned on me why it was that I had this obsession with things living on stones. It was quite a blinding revelation. Okay. Now here's a stone photographed in a local wood near where I live, and it's got a variety of things living on it. And as time passes, that variety of things increases, and more complex life forms appear on its surface. And it struck me that really mirrors very closely what has happened on the large planet-sized stone that we live on. In fact, further than that, it has struck me that the order in which things arrive on this stone, isolated in a wood, is very, very, very closely similar to the order in which science seems to have demonstrated that life has appeared on our planet. Of course, the difference is that life forms on our planet have evolved and developed over vast spans of time and actually changed from one species into another. But whereas on the stone in the woodland, things have arrived from somewhere else. Nevertheless, the sequence is very similar. Starting off with microscopic life forms, very, very simple plants, but plants which don't have seeds and reproduce by wind-blown spores and waterborne spores in some cases, mosses. Brave, tough organisms surviving in hostile environments and especially associated with dampness, water. That shows the spore-forming organs of a moss. Horsetails. Now these are the creatures, these are the plants that dinosaurs fed on. And again, they reproduce, they live in damp places. You can see the dewdrops on this horsetail, and they reproduce by spores. If you look at the back of the frond of a fern or bracken, you notice very distinctive spore-forming organs. One thing that distinguishes a fern, for example, from a flowering plant. Eventually, flowering plants appear on our wall, just as they appeared around about the time of the dinosaurs, quite late on in the history of our planet. Now, some people think that science, by looking at things closely, destroys their beauty. I think quite the opposite. Let's look closely at the flower of a buttercup. Okay? Now, a flower exists in order to produce seeds, in order to get those seeds fertilized. And when a buttercup flower reaches fruition, it ends up as a seed capsule like this. But this is a photograph taken with this microscope, with my own small digital camera, of a thin slice through a buttercup flower. Imagine a buttercup flower put through a bacon slicer, a very small bacon slicer, especially designed for slicing flowers. And you can see cells. Look closely at this picture. You can see cells, and you can see that the buttercup is made of a variety of cells serving a variety of functions and specialized for those functions. We can see the stem of the buttercup here with elongated cells in it. We can see that the green sepals, these are the petals here. The nectar is here. We can see hairs, special cells making hairs, in fact, on the sepals. We can see the stamens with pollen grains in. We can see the seeds developing here. And it kind of gives us a clear impression of the scale of cells in a buttercup. Let's look at a slice across the stem of a buttercup. Imagine a tree that's been chopped down or sawn down. You look at the cut tree trunk, you're looking at the cut trunk of a buttercup flower. Look at the beauty of the arrangement of the cells. Every single buttercup flower has veins running up its stem with exactly this configuration of cells, each designed for a particular function, which we could talk about, but we haven't got time. But the point I want to make is that one buttercup contains all that beauty. A whole field of buttercups staggers the imagination once your imagination starts thinking about that complexity, that beauty. So science doesn't destroy beauty, it reveals it. That's the tip of the root, actually, of an onion plant, not a buttercup, but it's exactly similar. Think about how hard it is to push a bendy stick into the soil. The stick bends and breaks. Imagine trying to push a hair-thin rootlet, soft, as soft plant tissue can be, into the soil. How does the plant do it? It's remarkable. The cells are dividing here. New cells are going in that direction and in that direction. In that direction they're differentiating. In this direction they're turning into balls of slime which are being sloughed off as the whole thing just grows down through the soil. And it can penetrate remarkably easily through quite densely packed soil. The whole system's amazing. You can see the cells, you can see the nucleus in each cell as a dark speck. It's been stained with stains which react with the genetic material and darken the nucleus in contrast with the rest of the cell. There we can see the cells more clearly. Again, these were photographed just with prepared slides on this microscope with a cheap digital camera on my sitting room table. As is this photograph, which very clearly, well, fairly clearly, reveals chromosomes. These chromosomes are engaged in a dance, which is remarkably similar to the dance we were performing last night, where couples, lines of people move together and then pull apart, partners separating and coming together again. That's the kind of dance which is happening here, which ensures that each daughter cell ends up with exactly the same set of chromosomes as the original parent. Here, that set of chromosomes is about to pull apart. Their two sets have just pulled apart, and a new cellulose wall is just forming between them. But I could have downloaded much clearer photographs from the internet, but as I said, this photograph was taken with this microscope with a cheap camera on my sitting room table. These things are real. And inside those chromosomes, in the heart of each cell, is the secret of life, is the secret code which determines whether that flower becomes a buttercup or becomes a dandelion. Encapsulated in that tiny seed in even tinier fragments of genetic material inside each of those seeds is the destiny of another plant in every detail. So all life needs is water and light and air. And this green stuff we've been looking at, that we saw in the chloroplasts, the first specimen, are able to manufacture food to feed the plants and to feed all the rest of us who live on the stone. Life is remarkably tough. Imagine surviving as a cell when the stuff that you're made of is freezing. Not everything does survive. In fact, nothing survives forever. Death is a part of life. It's an essential part of the process of life. It's not an accident. And death and recycling and renewal as the rotted away organism feeds other creatures, other organisms. Now then, what is it that breaks down the rotting plant? Well, I'd like to show you some of the creatures responsible for this process. Now, have you ever seen a vase of flowers which has been left to stand too long and the water in the vase turns milky? Yeah? That's caused by other single-celled creatures related to this one here. This particular kind is called Paramecium. And again, if you studied biology at school, you may well remember the name. Let's have a look at Paramecium. This stuff can be cultured by boiling hay in clean water and then letting it stand for a long time, and you can grow a culture of Paramecium like this. It's just like leaving a vase of flowers standing too long. Let's just pick some out of here and put it on the slide. Okay. Put a cover slip on to flatten it out. Let's have a look. Didn't need to do that. Nope, hold on. There it is. Switch the lamp on, would help. Wow, there they are. A little bit larger than some of the other cells we've seen, and they're swimming around quite actively. Now, it would be nice to be able to zoom in and look at these more closely, because each of these, instead of having a single flagellum or just two flagella, is actually covered with, I think, hundreds of much shorter but similar structures called cilia, Latin for a hair. And what I'm going to do, I'm going to try and slow them down. I'm going to play a very dirty trick on these creatures, because I'm going to put some goo in the water which will increase its viscosity, make it stickier, and slow them down. So let's try switching to this camera. Now, here's how we do it. If you hold some tissue paper, some absorbent paper at one end of the slide, it pulls everything in that direction. It sucks a lot of the stuff out from under the cover slip, including a lot of those creatures, unfortunately. Sorry about that. Let's put some of this stuff here and try to suck it back under the cover slip You have to bear with me here. This is slightly experimental. Okay, so you can see the moisture being sucked up into the absorbent paper, and I'm hoping that's pulling some of the gooey stuff under the cover slip. And then we're going to hunt for any remaining surviving Paramecium and see if they've been slowed down enough to see them better. Let's have a look. It would help to switch to the camera. Yeah. Please don't laugh. We must show some respect for God's creatures here. That one there looks hopeful. Right, okay. Please don't swim away. Never worked with animals. He's recovered. Let's find another one. Of course, you all wanted him to recover. Oh my goodness, what's that? Actually, I'm rather interested in finding out what that is. It's a tardigrade, absolutely. I would really like to find a source of these. These are remarkable creatures. They're sometimes called water bears, and they're really cute. Absolutely wonderful. However, it's a distraction from the present part of the story. Now, that's a multicellular organism. It's not a single-cell creature. It's actually smaller than the single-celled organism we're looking at. Right? Try and get your head around that. That is a very complex structure. We're going to come back to multicellular creatures later in the story. Let's go and look at Paramecium. With great reluctance, we'll leave the tardigrade. And let's see, because I've got got one more level of zoom yet, and we should. Oh, don't move away. Come on. Where you reckon it is? Oh, we've lost him. I'm sorry about this. Come on. There he is, he's up there. Now, come on. Get him in the centre. Using a new camera system here, and I'm just struggling. Oh. One more go. Come on. So I can see any familiar landmarks near that. Okay. Try again. Oh, there he is. Oh, it's like pantomime. He's behind you. Look at that. Now you can see the cilia. Yeah? Can you see them? Especially there. They're longer at the rear end. I think that's the rear end. I'm pretty sure that's the nucleus there. Okay? Amazing complexity in a single cell. Don't ever think of the sort of popular misconception that single-celled organisms are simple. In a sense, they're more complex than we multicellular organisms. Okay, Paramecium. Now, Paramecium survives by swallowing creatures like Euglena. Paramecium is not green. Do you notice that? Paramecium has to feed. It has no chlorophyll, and it feeds on small organisms which do have chlorophyll. Okay, now I'm going to show you this creature. Who knows what that is? Yes? Sorry? It's an amoeba, yes. I don't know. Maybe Amoeba proteus. I don't know the species. I just wanted to know what it was. Okay, that's brilliant. It's an amoeba. It's a single-celled organism which everyone's heard of, I think, just about, but very few people have actually seen. So let's have a look. Amoeba is a single cell which moves around by flowing like a giant science fiction blob, and worse than any kind of science fiction monster, it feeds by engulfing its prey. Imagine being swallowed, being engulfed by a sort of a tower block sized mass of goo that was flowing towards you, making horrible squelchy noises, flowed around you and then digests you alive in a little spherical blob inside its body. And this is what happens to the unfortunate green cells which are swallowed by amoeba. Again, creatures like Euglena. There are many other single-celled green organisms which creatures like this feed on. Okay, so let's hope we can find some. The scientific term for gunge is detritus. Okay, there's always a scientific word which sounds cleverer than the familiar word. I have to put just the right amount of water on the slide. Oh, sorry, you're not watching me. I have to put just the right amount of water on the slide so that the cover slip doesn't wobble around too much. It's a bit of an art to that. It's about right. Let's have a look under the microscope. Now again, you're going to have to help me here. You're going to have to help me find an amoeba. Oh! Looks as if we might be lucky. Can anyone see anything that looks like an amoeba? Yeah, that thing in the middle is a big blob of detritus. But these look very much like amoeba. Can you see the little green food organisms zooming around? Let's ignore those. Let's go in close. Now, we'll know if that's an amoeba if we see it flowing, if we see it moving. So let's watch out for that. Can you see any signs of movement there? Quite spooky with the sound of the wind in the background. Let's try zooming in closer. Again, just think about... Oh, golly, here we go again. Just try and think about the nature of life. This is really just a blob of protein dissolved in water and mixed with water and flowing. It's able to flow. And you just crack an egg into a saucer and look at the egg white. It's very similar chemically to this, but it doesn't do this, does it? And if you're a small green organism, it doesn't chase you. That creature is dicing with death. It's swum over it, I think. It's difficult to judge what's above and what's below under a microscope. Remember, this is three-dimensional. The amoeba isn't just flat shaped like that. It's got bulges upwards and even bulges downwards sometimes. It's decided to go that way. Can you hear it? There are traces of green inside it. Can you see? Where it swallowed stuff. There's a nucleus in there somewhere. I'm not going to try and point it out to you. A highly complex, granular structure. There are many, many unsolved scientific mysteries here concerning how this moves, how the whole thing operates at all. It's a remarkably complex thing. You know that inside your body you have blood cells, you have white cells and red cells. The white cells in your body look almost exactly like that. And they crawl around inside you, almost exactly like that, looking for dead cell material, pathogenic bacteria and so on to swallow. Okay? I'm coming to this point later, but we are very much part of this story. Now I'm going to show you another creature you may remember from your school days. It's called Hydra. And Hydra is also very common in ponds, but Hydra is multicellular. Hydra is a small relative of the jellyfish and sea anemones we see in the sea, but it's very tiny. It lives attached to plant stems. It has a sucker at one end of its body, sort of a sucker disc here. It's a hollow tube made of a couple of layers of cells, and it's got long stinging tentacles and a mouth here, which it swallows creatures with, and then digests them inside this hollow space, and then spews them back out again. It's only got one opening. These stinging cells paralyze its prey and pull it towards the mouth. That's Hydra, named after a legendary Greek monster. Let's see if we can find a monster. So, this is always tricky. I can see some in here. They are very, very small. I'll try and see if I can dislodge some and catch them. No. There I have one. Sorry, I'm going to use a different kind of microscope slide. I'm going to use one with a slight cavity on the top. Okay, I don't have time to be too fussy in selecting my Hydra, so we'll do with that one. Put a cover slip on. See if he's... oh, bother, there's a bubble there. Let's see if he's recovered from his ordeal sufficiently to expand. They contract when they're disturbed. Switch back to the camera. Now then, where is he? Seems to have a slight shortage of tentacles, this one. They normally have more than that. If you were a small pond creature, be very, very afraid. You can actually see the stinging cells, those little knobbly bumps on the tentacles. This is the lowest power of the microscope, but those little knobbly bumps on the tentacles are the stinging cells. They amazing things, amazing structures. They snap inside out and shoot out this long coil of you know those water snake toys? Ever played with those? Never mind if you don't know what I'm talking about if you haven't seen one. But they turn inside out as they move along, and that's how the stinging cells shoot out. I don't know if I can really catch them on a higher power. We can try. Notice it's green. Now what's going on here? Well, actually this species of Hydra lives together in symbiosis with a species of green alga. There are green cells of a completely different species living amongst its own cells. What is it doing? That thing's a bubble, by the way. That's just an air bubble. That strange UFO there. What is it doing? My word. What is it doing? I mean, science fiction films are pretty tame, really, aren't they, compared with stuff like this? They can somersault. They can move along by somersaulting. I think that's what it's trying to do. But it's pretty hard to somersault on a bubble. I have to keep moving because I've got so much to show you. We could stay with that for ages, couldn't we? But I have to move on. I'm going to show you some creatures which I caught in a tiny little pond in my garden at about 7 o'clock this morning. And the common name for these things is water fleas. The Latin name is Daphnia. And if anyone's kept tropical fish and bought live food from a fish and aquarium dealer, that's what you're dealing with here. They're very easy to catch your own, though. In ponds, what you do, you go to almost any pond, small pond in the summer, and make yourself a nice little net like this out of a very fine cloth, and just sweep it through the water in a sort of figure-of-8 pattern, just for a minute or so, well, a few seconds actually. All this reddish sort of soup collects at the bottom. You invert it into a container of water, and you've caught your own Daphnia, which is what I did this morning. Let's have a look at Daphnia. Daphnia is a multicellular organism, very tiny. It's actually in the same group of creatures as shrimps, crabs, prawns and lobsters. It's a crustacean. Let's see if I can catch one. Again, tricky with lots of people watching me. Let's see if we can catch one. A bit easier than the Hydra, I think. Oh, beauty. It's a big one. Sorry, I get too excited sometimes. Yeah, that's good. Oh, sorry, you're not watching. I keep forgetting to switch over. You missed all the fun then. I'm going to try and catch 2. Let's go for it. Flushed with success. Ah, I've got another one. Oh, there we are. 2 Daphnia. All the nicest invertebrates have names that sound like women's names. Now, I hope I've got the right amount of water in there, because it's another cavity slide, and I'm going to lower a cover slip onto the top as gently as I can. I don't want to kill the things, but I want to trap them underneath so that they're sort of held there and they can't move. And then we can look at them really clearly with a microscope, and then it gets really exciting. Okay, here we go. I think they're okay. They'll be fine. They'll be fine. They really will. They'll be released into the wild afterwards. I actually have to tell young audiences that. I mean, really, children get quite sensitive to this sort of thing, and quite rightly. We deserve to show respect to these things. Now that's remarkable. This Daphnia from my pond is diseased. I've never seen one like this before. It's covered with single-celled creatures related to Paramecium, but so this one. It's a sessile thing, a thing that fastens itself down with a stalk. Oh, I've got lots of things to show you here. Let's look at these things growing on it to start with. Let's try zooming in really close if I can. I'm not going to stay still, is it? Oh, he's up there. There he is. Oh, I can't keep him still. It's a creature called Vorticella. Okay, it's a can you see the things growing on the surface? They're like bells on stalks. It's called Vorticella. It's another ciliate like Paramecium, and it fixes itself down. It can swim, and it uses cilia to make feeding currents to bring its food to it. Let's go back to low power because we're not getting anywhere there. See his little face. I'm going to remove a little bit of the water. So you can watch me. Let's just suck out some of the water because they're able to move a little bit too much. They're really mean here. There we are. That should do the trick. Shave is upside down. See his little face, his eye. She. These are female. They're nearly all female. There's a a baby Daphnia there, at the back. Can you see it? Where's my laser pointer? That's a baby Daphnia. You can see the heart beating here. Can you see the heart? Yeah, okay. Still moving too much. Let's try the other one. This one's a little bit more obliging. That's better. That's better. There's the heart. There's the eye. Now the eye is a compound eye. It only has one. Sorry, Ben. It's only got one eye, this creature. And it's a compound eye with lots of lenses. And let's have a close look at the eye. For some reason, nobody knows why, the eyes twitch around constantly. And you can actually see the muscles which are twitching the eye around. Let's focus a bit better. The lenses are sparkling. They're highly refractive. Okay? See the muscle fibers here? Yeah? Now, what you can sometimes see is blood cells moving around inside the body. I can see a few. Not very clear here. He's got legs. It is a crustacean. Okay? Actually, limbs, I should say. Limbs. These are functioning as gills. There's the heart beating. Now, remember, this organism is made of lots of cells. Lots and lots and lots of cells. So just imagine the complexity of its organisation. This is its gut, and you can see the greenish, or it's become brownish, plant material that it's swallowed. And over at this end is his little bottom. And the heart beating. Quite remarkable. That's the mouth. That's the mouth there. Now, if I were to, you can, by all means, have a look at the Daphnia afterwards. I have a magnifying glass, and it's contained here. Just have a look at, marvel at how small they are, and at the complexity of the creature that we're admiring here. Okay, let's move on. So, I love to lie on my back in the long grass on a beautiful summer's day, like we've been having lately, looking at the small creatures that live in the jungle, in the grass blade jungle around my face. And think about, really marvel at the organisation of their bodies. Fearsome creatures. Really strange alien life forms. All these photographs were taken with a fairly cheap digital camera, sort of in and around my garden. Beautiful creatures. Cute creatures. Fearsome predators. Lovers. Robots. These ants are milking, these are farmers, these ants are milking aphids on the stem of a gorse plant. Okay, but eventually, on the giant stone that we live on, life forms which had previously only existed in water emerged from the water onto land. I wouldn't say dry land, it wasn't completely dry, but they emerged onto land. And the books always suggest that the first creatures that crawled out were a kind of fish with legs. That's quite wrong. Why should they crawl out? They crawled out to feed. But millions and millions of years previously, much smaller creatures had crawled out. There were mollusks, there were jointed leg creatures, spiders, okay, already existing on land. But eventually the most complex and advanced life forms of all, our ancestors, emerged onto the land. Would you like to see our ancestors, or a close relative of those early ancestors? Awesome, intelligent, complex, the first amphibians. Okay? So now I just want to demonstrate the connection I referred to before between us and this story. And at this point, what I want to do is to show you some living human tissue under the microscope to show that we're of the same stuff. And in order to remove some living human tissue, I require a volunteer from the audience. Okay, lady at the back, her hands shot up. Thank you. Would you like to come up the steps here and stand here? Thank you very much. I'm so sorry, I can't remember your name.
Judith.
Sorry?
Judith.
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 Yeah.
Okay, Judith. Okay. Now then, here's what we're going to do. Let's put the slide on here first and switch to this camera. Now, using this implement, okay, we're going to remove some cells from the lining of your mouth. And what I'm going to ask you to do is to put this teaspoon inside your mouth and rub it 3 times against your cheek, against the lining of your cheek. 1, 2, 3, inside your mouth like that, okay? And a little bit of spit and a large quantity of cheek cells will be on the spoon, and then pass me the spoon. Now this spoon is very clean. It's been nowhere near these creatures here. It's been right over there. It's a clean teaspoon on a clean tissue. It's absolutely fine, okay? Thank you. 1, 2, 3. Okay, let's have a look. Let's see what we've got. That's absolutely brilliant. Yeah. Okay. Can you give you this a big clap? Go and sit down. Thank you very much. Okay. Now, please don't say yuck, because that will offend our volunteer. Okay. But we're just going to rinse some of this sample onto the slide. Absolutely wonderful. That's good. A little bit too much water. It's always a shame to do this demo after lunch. Actually, the best way to remove this is using the tissues before I put too much water. I need to get plenty of water off this because I don't want this slide to float around. Let's burst that bubble. Great. That's it, got it. A cover slip. A bit clumsy. Remove a bit more water. Okay. I don't want this to be floating around too much because we're going to go on to high power of the microscope. Let's have a look. Okay. Now, those are the cells. We can see the cells. There's an awful lot of them. Each of these is shaped almost exactly like a fried egg. Each cell is specifically designed to suit its function, and the function of these cells is to form a nice, slippery, lubricated lining inside your cheek. And these flat, fried egg-shaped cells can slide one over another very readily. But you can't see any detail. Let's go in closer. That's very good. I'm going to try and choose one cell which clearly shows the shape. Let's choose that one up there. Focus. And again, I'm going to zoom in closer still. Good. It's a bit like Etch-a-Sketch, turning these knobs in the right direction. You know that children's game where you draw a picture by turning two knobs. And there we see a small piece of Euglena, and we can see that the cells that she is made of are just the same kind of thing that we've been looking at throughout this story I've been telling you. So the remarkable thing is that we're part of this story. We're made of the same stuff, and we're inseparably connected to this story. We share the same basic needs, and we live on the same planet. And there in the middle is the nucleus, containing the secret of life that makes Euglena different from everybody else. Every single detail needed to make Euglena unique and to control every aspect of her body, of her life in a sense, is coded inside that tiny, tiny, tiny speck in the center of that tiny cell. There it is, shining like a jewel in the center. Thank you very much indeed. That's the end. Thank you.